Today's guest is Amos Gdalyahu, and very happy to have him here today. Amos completed his bachelor's degree in life sciences at Geffen and continued to the Weizmann Institute for his master's and PhD, in which he worked on molecular mechanisms of neuronal migration necessary for cortical development and their involvement in developmental diseases. Amos then went to a postdoc at UCLA with Josh Trachtenberg. There he worked on the effect of sensory fear conditioning on cortical coding in the somatosensory cortex using in vivo two-photon calcium microscopy and novel learning paradigm. Amos then worked on cortical coding to understand mice models of autism. This was with Josh and Dan Geffen. Very recently he returned to Israel and joined the lab of Pablo Glinder at Tau, where he's using other two-photon microscopy tools to reveal mechanisms of neurovascular coupling and neuronal coding. Please welcome Amos. Thank you very much.
Thank you. Thank you, Shai, for this kind introduction. Thank you all for coming. I'm really happy to be here, finally. You hear me? All right. So I want to start my talk with my motivation to do my studies. And I think it is said best by the words of the Dalai Lama. Health is the suffering of psychiatric diseases. If you could really see the pain around, you would not be able to sleep at night. So I think this is common for many of us. We came into neuroscience because we were interested how does the brain work, and later on you see the suffer around and you really want to do something to solve these awful psychiatric diseases. But the question is how? And obviously this is not going to be the work of one lab or dozens of labs. It's going to be the work of dozens of thousands of labs working for decades around the world. And eventually, by that, we'll get there. And I want to contribute my piece. And of course, there are many different levels to study cognition. You can, and eventually to cure psychiatric diseases. You could you could work in the behavioral level only and see how does an animal behave, what are the rules of behavior, theory of behavior. You could dive in and map where cognitive functions are located in the brain and do this geography in the brain, as we see here. Or you could dive even more into the molecular or biochemical level. Here we see a synapse. This is the postsynaptic structure, the spine, and you can study what happens after a cognitive task to the proteins, where are they translocated, what proteins are expressed, and so on. I chose, and it's an entirely subjective decision, I chose to work at the circuit level. And this is here, shown here. What do I mean when I'm saying circuit level? Circuit, network, people use these two terms. They mean exactly the same. I mean neurons that reside near each other, and together they mediate the function that is carried out in this brain area, the cognitive function. And I want to understand how do these neurons mediate this function. And the reasons I choose this level, I think, is the following. One, it is kind of in the middle, and it allows me to integrate all the different levels from behavior until the molecular level in my studies. And second, these days we have excellent new tools that allow us to answer these questions in vivo and to reveal answers for questions that we dreamt to answer before, and now we can. So I'm focusing on two fundamental properties of neural networks in vivo: connectivity and activity. Here you see this structure. This is a dendrite, and this spine. Exactly what we see here. So each spine is a synapse, and think about it. This is like 2 microns each of these spines, and we am able to see them in vivo. And the other thing that we see here are many neurons each one of these green dots, a neuron, and I can see them talking inside the brain, all of them together. So in order to understand behavior, I believe that we need to understand connectivity and activity. And we are now able to do this in vivo. This is a revolution in neuroscience. So in my studies, I focus on adaptive perception. What do I mean when I'm saying adaptive perception? I mean, let's say you are in the jungle, you want to hear the lion before the lion hears you. You have to be very sensitive to the lion, so it makes sense that you would perceive the sound of lion from very long distance. When I'm here, in Israel, I don't need to be so sensitive to the sound of lion. For sure, the lion would catch me much before I hear him. But I'm very sensitive to my cell phone ringtone. And if I'm buying a new cell phone, after some time I'll be very sensitive to my new ringtone. So my perception is continuously modified by experience. This is adaptive perception. And I always have in my research two labs. One is in health, in this case learning, and the other in disease. And these two levels, even if they are separated from each other, they inform each other. And in order to understand and to solve disease, you must understand the healthy brain, and this understanding allows you to go into the disease. And I hope you will see this today. So in learning, I'm asking, how do neuronal networks learn? And specifically, how does learning change network activity in the sensory cortex? Okay, and here I'm going to tell you, to show you activity, the activity of the network in vivo after learning. Okay. In disease, I'm going to tell you what neurological processes go awry in psychiatric diseases. That's my general question, and of course I'm going to focus on much narrow question today. And because unusual sensory behaviors are common in autism, I will focus on autism, and I'll ask how autism-related mutation affects the connectivity in the sensory cortex. So we'll see both activity and connectivity. But let's start with network changes in sensory cortex after learning. So here we see a tonotopic map of the primary auditory cortex of rats. Each color here indicates the frequency that activates this brain area best. So, for example, here in gray, you see 4 kilohertz. This is in naive rats. After learning that this 4 kilohertz is a signal for reinforcer, you see the expansion of this frequency. So that's the effect of learning in primary auditory cortex. And naively, people thought, All right. So hypothesis one was that learning increases the number of neurons. Why? Because when people see this expansion, naively, they thought that all neurons inside each polygon have the same activity. If we have more polygons, we have more area, we have more neurons responding to the stimulus. Of course, Adi has shown us, Adi Mizrahi here, shown that there is huge heterogeneity in the cortex, but still that's the assumption. We all learned that in basic neuroscience, that this expansion, and the assumption, the hidden assumption was that after learning, there are more neurons responding. And that, I think, was mainly popular within a neurobiology department. In the psychology department, people had the exact opposite hypothesis, which said that learning reduces the number of responding neurons. And this is shown here. Again, so each circle here indicates a neuron. The red ones are the ones that respond to the stimulus before learning and after learning. And this idea is based on behavioral studies. For example, Pierce and Hall, the negative transfer model. Merav Achisar here also contributed for this. So we have two exact hypotheses, and I wanted to tease apart these two options and to know which one is the correct. So now I have the tool. I have this possibility to image network activity inside the brain and to answer this question. And let's see that. So here I'm going to show you, this is low magnification. You see here many neurons that are talking with each other within the brain of a living mouse under the microscope. Here, this is from UCLA, this is Tel Aviv University. Here in red, in green you see neurons, in red you see blood flowing within blood vessels because this is a live animal, bless you. So we are able to see network activity in vivo. Not only that, we can also see neuronal identity. Here we see one inhibitory neuron and one excitatory neuron inside the brain. So, and by the way, they're totally uncorrelated here. So we have now a tool that allows us to answer the question. So let's do it. But there is a problem. My tool, two-photon microscopy, requires optical access to the memory trace. And most memory traces are in places that are optically inaccessible, if it is the hippocampus or amygdala. And I was talking so far about sensory cortex, and even in the sensory cortex, the same, like auditory cortex, I did it, but it's very difficult, very, very lateral. It was considered, before I did it, it was considered impossible. Olfactory, hey, olfactory, you have to remove half of the face to get there. If it's taste cortex, again, you have to remove the eye. Very complicated surgeries, very unpleasant to everyone involved. So I wanted to use a different modality. So I decided to use the whisker modality. And here you see whiskers in action. So whiskers are a very sensitive device that rodents use to navigate around. They are night animals. They go in tunnels. They rely a lot on the whiskers as if you were going in a dark room and trying to find your way around like that. So that's the whiskers. Very sensitive. Information from the whiskers arrive eventually to the primary somatosensory cortex, you see here. And if you cut a slice in layer 4 of the somatosensory cortex and you stain it for the activity of the enzyme cytochrome oxidase, you get this typical pattern of staining in which each dark area is called a barrel. Each barrel deals with information from one specific whisker, so we get a functional map in a slide. And this feature has facilitated research in this system, and we know already a lot about this system. So, and this gives the term barrel cortex to that part of the primary sensory cortex that deals with whiskers. Here you see the barrel cortex. You see this typical organization of the map of the whiskers, and you see the location. Also, you see all this pink is the primary sensory cortex, and the barrel cortex occupies about 1/3 of it, showing its importance. And you see it here, whereas auditory cortex much more lateral here. So primary, the barrel cortex is optically accessible. Therefore, I decided to develop a new associative learning for the barrel cortex. Because associative learning is a sensory-sensory procedure, it also makes sense to look at the barrel cortex after associative learning. And the way I did it, I wanted to associate a stimulation of a whisker with a food shock, and then the mice would learn the association between them and would show fear in response to stimulation of a whisker. So how did I do it? I glued a small metal, like 1 millimeter, on one specific whisker, as you see here. Then I placed the mouse with this metal in the bore of an electromagnet that you see here, a side view. Here it's from the top. When I delivered current into this coil, it created a strong magnetic field. This magnetic field moved the metal with the whisker according to the right-hand rule, up or down. So I delivered that current in a frequency of, in whatever, 8 hertz, and this caused that frequency of stimulation of the whisker. When I wanted, I delivered also current into the floor, which was in the center of the electromagnet. This gave the mouse a food shock. And here you can see a mouse getting a food shock. Oops. All mice are freely behaving here. They walk around, then they get this visco stimulation, they get the shock, and they learn the association. The mice didn't like to have a metal on the whisker, so they would try to remove the metal, and if they couldn't, they would pull out the whole whisker and would terminate my experiment. So I placed on them a collar that prevented them to reach this whisker. They hated that, but after some habituation, they were totally fine with it, and I was able to train them. So I had two groups of mice. Group 1 had 5 whisker simulations, each one of 30 seconds, and each one was followed immediately by a foot shock. So here they had a foot shock immediately after a whisker simulation, and they're supposed to learn that the whisker simulation predicts the foot shock. The second group received both stimuli, but explicitly unpaired. First they got 5 shocks, and then they got 5 whisker simulations, so none of the shocks is predicted by whisker simulation. Then I wanted to see if they have learned and formed the association between the two stimuli. For this, I placed the mice in a different context, so it was the same electromagnet. I had only one. But I put another tube inside, so it totally changed how it looked, and you can see it here, different flow, all the walls were different. And now I'll show you, and the reason I did it is because I didn't want to see context fear. I wanted to see fear that is induced only by the whisker stimulation. So I'm going to show you now the behavior of 4 mice. These three were trained with the paired protocol. This was trained with the unpaired protocol. The mice would walk around. When you see here a white circle, it means the whisker is being stimulated. So they're first now. So they go, they explore. It's of course fast forward. And after 4 minutes of baseline, they would get this stimulation. So this, I didn't say, when rodents are afraid, they freeze. This is very conserved behavior across evolution. Even in humans, many times victims of abuse or domestic violence are asked, why didn't you do anything? Why didn't you scream? Why? And they say, I was paralyzed. I couldn't move. It's the same. It's something, or in Hebrew, we say, likfo mipachad. It's exactly the same. It's very conserved behavior. So I hope, as I was talking, that you were able to see that. These were typical best students. The group data is here. So here you see a percent of freezing in paired mice and unpaired mice. This is during baseline. And during the time of whisker stimulation, my conditional stimulus, the paired mice froze significantly more than the unpaired mice, meaning they formed an association between these two stimuli. And this memory is very long-lasting. I tested the mice even 1 month after learning, and still they remembered very well. This is a totally new behavioral task. So I had to do a lot of a characterization of this task. I measured generalization to a different whisker, discrimination between 2 whiskers, one paired, one not paired, trace conditioning, effect of short context habituation, and lots more. If you have questions and you wonder about some behavioral parameter, just ask me. Probably I did it. But for the sake of this lecture, I want to continue to my basic question. How does learning change the number of responding neurons within the cortex? So I have the behavior, I have the microscope, and now I can image. But I do the surgery, and all I see is blood vessels. Where are those barrels? Where is the barrel that I trained? So to find this barrel, I used another method of brain imaging in live mice. This is called intrinsic signal imaging. It was invented by Amiram Greenwald in '86, and the idea here is the following. You have a mouse under a camera, and you stimulate the whisker periodically. The area in the cortex that is activated by this whisker stimulation consumes a lot of oxygen, and therefore the blood there becomes darker. because it is darker, it absorbs more of the red light that we shine on the brain, and less red light is reflected to the camera. So in activated areas, in time of the activation, there is reduction in the signal, whereas in other areas that are not activated, there is no difference. These are very small changes, but we can see them. So I use an analysis that hasn't been done before in barrel cortex. It's based on Fourier transformation. So I did some controls to show that it really met the whiskers well. Here I deflected a whisker B1 or C1 or D1. They have names, by the way. And you can see that the response changed. And also I injected a virus to what I thought was a barrel D1. And later in a slice, indeed I found the expression exactly in D1. So my imaging worked really well. And now, so I have my behavior. I know I have the windows. I have intrinsic on imaging, and then I knew where to inject calcium-sensitive dye. So you can see this is exactly the spot. And now I injected calcium-sensitive dye, and you can see many neurons that took up the dye and are labeled in vivo. So I wanted to see one group of neurons, one network, across time, and to see the response to the whisker stimulation. So again, I'm going to show you a movie in which whenever you see a white circle here, it means the whisker is being stimulated. And by the way, neurons are in green, astrocytes are in orange. And if you look on this neuron, you will see that each time the whisker is stimulated, this one is responding. You can actually look at this one and know when the whisker is stimulated. So this is going to be 10 times. Oops, sure. All right, and this is the same field of view. And here, each neuron got a number, and we can see the traces of these same neurons here. This is the neurons I was talking about, and you see its responses. And obviously, you see that there are huge variability between the neurons in the response. So here I have a schematic representation of a network. Each circle is a neuron, and the color indicates how much this neuron was activated in response to the whisker stimulation. So the average, if you average the responses across 10 trials, you give this value here, you give that to each neuron, you get this map. Based on spontaneous activity, I determined a threshold above which I considered a neuron as responsive and not just spontaneously active, right? Because the neuron could fire at the time of the whisker stimulation, but maybe it just happened to fire as spontaneously. So I had a threshold, and then I was able to quantify my movies. Here you see the result. So this is cumulative percent. The paired group is in solid line. So, and this is the percent of responding cells within the field of view. So in the paired group, we see that about 50%, up to 50% of field of view had about 40% neurons responding in them. But in the unpaired group, 50% of field of view had about 80% of responding neurons. So, and this is also summed up here, and you can see the reduction, significant reduction in the paired group. So this is across 10 trials. There is reduction in the number of responding neurons. What happens in a single trial? In a single trial, the neurons, we could have the same or not. It depends what is the fidelity in which the neurons respond. So I quantified also responses to a single trial and found the same. So the conclusion is that associative learning reduces the percent of responding neurons. So far, I have looked at each neuron in very binary way. Either it's active or not active. But there is more information in this data. We can look now on the magnitude of response. So within the responding neurons, how much was their average response? So I did that, and I found that within the responding neurons, in the paired group, there was more response. So taken together, associative learning causes less number of responding neurons, but the responding neurons increase their magnitude of response. So we see here a specialization in the somatosensory cortex for processing of a conditional stimulus. This specialization improves perception, I claim, improves perception of a CS, of a conditional stimulus, because of less crosstalk between neurons. And these effects were specific to associative learning. I'm not showing it now, but I did it all with another task of non-associative learning, and I found a totally different results. The number of responding neurons was not changed at all. You have a question there? Yeah.
First, do you think it might be specific to ear conditioning, or did you try anything with reward and now?
Excellent question. So within the associative learning, I did not write appetitive versus negative. There are reasons to think that there could be a difference, but I don't have any data for it. What I know is that non-associative learning causes effect too, but they are very different.
And even more than that, when you're talking about more neurons firing or less neurons firing, there are networks with increased inhibition, networks with reduced inhibition, and it's hard to say like that. Were these only pyramidal cells or?
Yes.
I mean, it sounds a bit too simple, you know?
Well, this I don't know. This is the data, but this is what I see. But to dive in, I agree with you. It's an excellent point, and I'm going to go into it later on for the inhibitory neurons, excitatory neurons, totally. This was my paper was the first paper actually, one of the first. I think it was actually the second that looked at the effect of learning in vivo in using calcium imaging. So of course we can go more and more, and yeah, there's lots more to discover, and totally I intend to do that.
One more question. Do
yeah.
these neurons respond to other stimuli except the whiskers?
These neurons?
Yeah.
I did not check that.
Noise?
Yeah, I did not check it. This is primarily, this area deals with whisker. Ron Frostig in USC, I think, or Irvine. Anyway, Ron Frostig has data that shows that even neurons that are in the barrel cortex respond to auditory stimuli. So, but I didn't check it.
Right.
And another thing for your question, my data since then were already replicated by other labs in other systems. So I don't know about all tasks if they have the same effect, but at least in few more it has been found the same, including a brain-computer interface kind of. Yes?
Is this same neuron or like two different populations?
Is they paired and unpaired?
Before and after?
No. Here I used synthetic calcium dye, and with synthetic calcium dyes you cannot do chronic imaging. That was since then. Now we have great genetic tools, and now we can do it chronically. But I had to still compare paired to unpaired. Yeah. All right. So I want to move on to my disease part. So I've told you so far about the network and about activity, and we know that unusual sensory behaviors are common in autism. What do I mean with that? I mean that one person with autism might not be responsive to touch. You touch here and they don't do anything. They don't look back. One person with autism would love to stare on blinking lights. It would be very fascinating for them. Someone else with autism would hate to go into a room with a lot of noises or to a mall. It would be enormous, very unpleasant experience. They would run away from the mall. So these are all very specific behaviors. All are related to sensory behaviors, but there's nothing that really puts them all together except abnormal, unusual sensory behaviors. So it's not that people with autism don't hear or don't see or don't feel touch. They do, of course. But there is something more subtle in the way the sensory information is maybe processed or something in the circuit is different. So we thought it could be a problem of connectivity within the sensory cortex. So I wanted to see how does an autism-related mutation affect the connectivity between neurons in the cortex. So
if you touch a person, he feels but doesn't respond?
not necessarily. It's not like 100% of the people. It's not part of the definition. It's not one of the two components that are now needed.
But he tells you, I felt, but I didn't respond?
So in people that have autism, it's very common to find unusual sensory behavior. It could be that, it could be something else.
Yeah, but the particular person, when you touched him and he didn't respond, did he say, I felt it, but I didn't respond? Or he said, I didn't feel anything?
It's a good question. I actually don't know. It's a good question. I don't know. Yeah. So, few synapses less, few synapses more. Why should we care, right? I mean, each neuron has like 1,000 synapses, few more changes. Who cares? So here we see a dendrite in blue and several axons that pass nearby. Out of 10 axons that pass close enough to this dendrite and could form a connection with this dendrite, only about 3 actually do form a connection. So this means that the system is that only a minority of the possible synaptic connections actually exist, and that synaptic connectivity is highly regulated. And if something is highly regulated, it's probably important, and it means that changes in connectivity could have major consequences because the system is so not saturated. And indeed, in recent years, it has been shown that changes in connectivity mediate learning, extinction, and memory. So, I wanted to see if an autism-related mutation affects the connectivity, and some of you may ask what a mutation has to do with autism. So autism is actually highly genetic disease. Here you see the chance to have autism within twins. If one of them has autism, this is when they are identical twins, and compare this to non-identical twins. So we see that identical twins have much higher risk to develop autism. So it means it is very genetic, and compare even non-identical twins, like siblings, to the normal rate of autism in the population of about 1%. So we see a major effect of genetics on each of the endophenotypes of autism and autism itself. So this means that now we can identify genes and create mouse models of autism. And I used a mouse model based on a CatNap2 knockout, and we think this is an excellent model for autism because of the following. In humans, a loss of function of CatNap2 causes autism with more than 70% penetrance. This is the highest penetrance of all autism-related genes. And also other mutations of CatNap2 with lower effect are correlated with endophenotypes of autism in the healthy population, like the age of speaking the first word, for example. We show that these mice that have a knockout of CatNap2 have all the behaviors that characterize autism. Problems in social communication, repetitive behavior, and the related problems like abnormal sensory behavior. And this was in a cell, and this mice also responded to a drug as the same way as people with autism do. This is risperidone. It corrects their repetitive behavior, but not the social or the communication problems. So I used this mouse model to ask, does an autism-related mutation impair neuronal connectivity in the sensory cortex in vivo? And here you see one typical dendrite in a wild type, and here in this mouse model. And This is the quantification of the number of spines per mouse or per cell, and you can see that there is a significant reduction in the number of spines in the knockout, meaning there is less connectivity in the knockout. How does this reduced connectivity arise? So the power of this technique is a chronic imaging, and then you can really see why things happen. Because to look at connectivity, we could do also in a fixed brain tissue post-mortem of people, right? But in people, we can't follow up, it's already dead. Plus, in people, it's very heterogeneous population, whereas in mice, all of them have the same mutation. So we have the ability to answer this question, the benefit of mice here. So I followed about more than 4,000 spines in vivo in 3 times. So I had a window. Here you see this neuron that's located here, and I followed this segment. So first of all, I looked at spines that were present. This indicates a spine. The black indicates the dendrite. Spine that's present here and was not present here. I quantified that, and I found an increased spine loss in the knockout. Then I measured spine gain, so spine that appeared at day 4 but were absent at day 0. And here I found no change. So together, more spine loss and unchanged spine gain explain the reduced connectivity. And now you can ask a new question. What causes the increase in spine loss in CatNap2? There could be 2 different problems. One problem is of maintenance. This is, let's say, in a knockout, we have a spine that was present for 2 days, so it's stable, but then it was lost, whereas in the knockout, in the wild type, it would be still present. That would be a problem of maintenance. Other possible explanation is a problem in stabilization. Let's say we have a new spine that was formed here, and in the knockout, this new spine fails to stabilize, whereas in the wild type, it would. So to distinguish between these 2 options, I added another time point here, and it allowed me to distinguish between them. So first of all, spine maintenance is unchanged. So spines, the percent of spines that were present in here and here was not different at day 11. But spine stabilization was very severely reduced, about 60% reduction in the stabilization of new spines in the knockout. Again, this is per mouse, per cell. So altogether, in CatNap2 mouse model of autism, I find that stabilization of new circuits is impaired. And this means that in general, in the healthy situation, stabilization of new spines needs CatNap2. So again, this connection between the healthy situation and the disease. Each one, they both inform each other. Here we see the life of a synapse in wild type. There's a dendrite, it goes a philopodia. 90% of these philopodias are lost within hours. Like some of them remain, about 10%, and then there are new spines. Most of these new spines disappear within days in activity-dependent manner, and few of them become young spines. This happens in the wild type, but does not happen in the knockout. Then these young spines have to continue to mature, to get the protein translation machinery, and eventually they become mature spines that also have to be maintained, and CatNap2 does not change here. So I told you so far about 2 studies, 2 distinct studies, not related, except both of them are related to adaptive perception. One of them dealt with activity, one of them with connectivity. Of course, I did the vice versa. I also looked on connectivity after learning. This is in a paper we recently sent to Nature Neuroscience, and we found here, we can find that new spines that are formed during learning are not formed randomly, but they are formed in hotspots along the dendrite, in clusters, and also the loss is clustered, and the rate of synapse formation and loss before any learning is a great predictor for future learning of these same mice. And I also looked on network activity in this mouse model, and I found reduced synchronization. But I want now to continue to my future plans here. So, no? Yeah, so here, if it, oh, why it's not playing? Oh, all right.
Is this an autistic person?
What?
Is this autistic person?
I don't know. I don't know. Don't know him.
Is an autistic actor?
So here you have a half-year follow-up of the same dendrite, by the way. All right. So first question that I want to answer, is there a memory in inhibition? And this kind of communicates with your thing. So the basic model of inhibition after learning is shown here. Here you see several excitatory neurons around one inhibitory neurons. And the idea is that after learning, this inhibitory neuron change its activity, but and it allows the excitatory neurons to change, but eventually the inhibitory neuron would go back to its normal, to what it was before, and there is no permanent change in inhibition. My findings suggest a different model of inhibition, and I'm not talking here about change in the amount of firing in the soma. I'm talking about a change in the projections of these inhibitory neurons. To remind you, I find that after associative learning in the barrel cortex, there are less number of responding neurons, but the ones that respond stronger. Taken together, I predict that some of the projections of the inhibitory neurons would become stronger, so they inhibit the excitatory neuron stronger, and we don't see this neuron responding anymore. And some other neurons are inhibited less, and this is why the magnitude of response increases. So even if the rate of firing of these neurons would go back to what it was at the beginning, we would still have a permanent memory in these neurons. And this means we have a memory inhibition. I have some unpublished data that support this idea, and here I actually measured the responses in my networks in using different stimuli, not only the conditional one. And as I increased the intensity of the stimulus, I found more and more, this is the percent of responding neurons. As I increased the intensity, I found more and more neurons responding. And so, because, and this kind of fits with an idea of inhibition. So if the neurons are still in the circuit but are inhibited, if you give them a stronger stimulus, you can overdrive the inhibition. So I want to test this model using genetic tools that we now have, the Twitch.
Okay, on x-axis over there, should that be hertz or decibels?
It has. So I deflect a whisker in different frequencies. So you are used to a tone, right?
Well, okay, this is whisker stimulation.
This is whisker stimulation.
Faster stimulation.
What?
It's faster stimulation.
It's faster and more, like if instead of giving a train of 5, I would give a 10 or 40. So it's stronger. Yeah, so I want to see the projections of this inhibitory neuron before learning and after learning. And this I'm now able to do by targeting genetically encoded calcium indicators into the inhibitory neurons only, and look at the axonal boutons, both their presence and their activity before learning and after learning. Another plan that I want to conduct is shown here. What was it? How does the spine deficit in knockout, in the CATNAP2 knockout relate to behavior? So one explanation to my findings is that there is insufficient sensory input that causes this problem. So to check this possibility, I want to saturate the sensory information that mice get and by placing them in this Mardi Gras cage when they have constant stimulations of the whiskers all the time. They cannot avoid it. So you totally saturate. And indeed, in wild-type mice, this procedure has already been shown to increase the stabilization of new spines. So exactly the process that I see is defected in these mice. So if I saturate this sensory information, would it rescue the effect? And by the way, we know that in the therapy in autism of intensive behavior is helpful. So,
what do you mean therapy by intensive behavior?
Like ABA, for example, like 40 hours of intensive behavioral therapies, someone that works with the person for 40 hours a week, for many weeks, there is improvement in performances.
So it's not permanent stimulation, but
Right.
interaction.
Yeah, sure. Another thing, and we know that the stabilization of new spines is necessary for learning, and this is defected in these mice, so I would want now to check the sensory learning in these mice using the task that I developed. So now I want to move to another direction of my research that unites these two studies that I have talked to so far. And does rewiring in the prefrontal cortex mediate flexible behavior? So far when I talk about the sensory cortex, and the sensory cortex is great because you know exactly what you put in. I know exactly what is the stimulus, I have total control over it. But there is a problem. It's very far away from the behavioral output. So when people ask, okay, but how do these changes correlate or affect behavior, you have answers, but it's a lot of hand waving. So I want now to move closer to the behavioral output, and I'm going to do that in the frontal cortex and using flexible behavior. And what do I mean with flexible behavior? So let's say you are an animal. Animal encounters sensor input, where it is here. Normally, the response to this sensor input is behavioral output 1. This is the track that the animal would do. Then the animal learns something about this sensor input. Let's say it's dangerous, and it needs to change its behavior. So it learns a new behavioral output, and this is shown here. So it formed a new rail. This is the learning. After that, it learns that this stimulus is not dangerous anymore. So now it goes back to behavioral output 1. It has not forgotten behavioral output 2 or 1. It just flexibly moves between these two outputs based on the circumstances. So this is behavioral flexibility. Across life, we learn many different responses to the same stimuli, and we decide what to do based on the context. So this is the process that I want to go in. This is what I call a flexible behavior, and this process is defected in almost any psychiatric disease that you can think of. If it is addiction, anxiety, and of course in autism. So again, these two things are highly related to each other. And here, I want to look at a brain area in the prefrontal cortex that Wen Biao-Gan has already shown its involvement in fear learning and extinction. So here we see a dendrite and spine on it before learning. After he trained this mice that a tone is a predictor for a foot shock, he saw elimination of spines in that area. After he did extinction, so he gave only the tone without a shock, he found that spines were kind of reborn at the same area where they were lost before. And he took it to say that extinction partially erases the first learning. But there is huge literature in psychology that shows that extinction does not erase the first learning. So I want to suggest a different interpretation to these results. My interpretation is that these changes here are not related to the memory. They are related to the behavioral response that the animal chooses to do. So this is the flexibility that we have in the system. And now I'm going to tell you how I'm going to test my hypothesis. So, first, there's absence of spine stabilization in the prefrontal cortex of wild-type mice cause inflexible behavior. So I know how to cause instabilization of spines. I found that if that CatNap2, this is exactly what it does. So using the study of disease, I now have a tool that allows me to decipher a basic mechanism in health of flexible behavior. So I predict that if we block this formation, the stabilization of this spine, the extinction would be impaired. And then, of course, we can go to other tasks of flexible behavior. Second hypothesis that is going to prove second experiment that is going to prove the hypothesis, there's overexpression of CatNap2 in CatNap2 knockout rescue flexible behavior of these mice. So I would restore CatNap2 only in that brain area. So a window over the prefrontal cortex is a bit tricky, but together with Pablo Garcia Junco Clemente, now I'll call him Pablo, we figure out how to do that, and it works. I can show you also a calcium movie from there. And so far I was talking about the input into frontal cortex. I was looking on spines. Now I want to also to look how the output from the frontal cortex affects behavior. So what I plan to do here is to inject a virus that encodes a genetic indicator of calcium to inject it into frontal cortex and to look at the axonal buttons within motor cortex. So the projection from frontal cortex to motor cortex. And I predict that learning would cause a change in the projections from frontal cortex to motor cortex that enables the change in the motor output. So altogether, my interest is to reveal the cellular level of behavior. I've told you that in adaptive perception, I developed a new associative learning, and I found that it reduces the number of responding neurons, and I want to check how inhibition is changed during learning. I also showed that in autism or autism-related mutation decreases stabilization of new spines in the primary sensory cortex, and I want to see if I can rescue this effect by behavioral manipulation. And I've told you about a new direction of flexible behavior where I want to test my hypothesis and using my finding in disease as a tool to see if stabilization of new spines really necessary to have extinction, and if the output from frontal cortex to motor cortex is changed during learning, and obviously eventually we want also to look at the network activity in frontal cortex before and after learning. So I want to finish with thanks to all my colleagues and the people that have worked with me, and thank you all for listening.
We have some time for questions, Steve.
I didn't get exactly how you see that one area is projected to another area. Do you see just by, you say that activation, like serine activation, or at the same time, or sequence activation, or just by correlating the neural activity, or by synchronization, or do you look on the connectivity?
You mean my future plan when I want to look at the projections from frontal cortex to motor cortex? Yeah, so what I want to do is to express in the frontal cortex a, a virus that encodes calcium-sensitive protein, and this is a protein that is threat-based. So all the time I see the fluorescence, so I can see the structure of the neurons, so all the axonal butons, and I can also see at the same time the functions. So I will be able to look at both the structure and function of the butons pre and post learning. Does that answer your question?
So actually you see the connectivity, okay, you see the projection by the connectivity that you see according to the fluorescence?
Right. I see actually the neurons, I actually see the projections.
I see, but here you don't see the rhythmical connectivity. Like if they speak like in the same rhythm, the same, I don't know.
Temporal contingency.
You mean as a population?
Yes.
No, that's not my, I mean, that is something that we can explore. It's definitely interesting, but my aim right now was to stay in the cellular level and look if the projections themselves are changed. I mean, eventually this may lead to more global effects that you can see in the population level. Well, yeah.
You said something about hypersensitivity in autism on one hand, and then you said something about decreased activity, which is somehow correlates with instability of spine. So it seems to be kind of going together. But if you have hypersensitivity.
So first of all, I mentioned hypersensitivity, but also hyposensitivity. Both can happen even at the same person. What I say is very common is unusual sensory behaviors. It doesn't need to be hyper or hypo. Now, is that, yeah? Okay. Yeah.
In the beginning, you presented a slide that showed some somatosensory cortex, and then you showed that if you, I don't know, make stimulus in 4 hertz, the area of 4 hertz
Yeah.
is bigger. How is it modified to your?
Yeah. Excellent question. So, what you are saying, there is evidence for expansion, and I see less number of neurons. How to put them both together, right? So, to get map expansion, you need very intensive behavior. All the studies that have shown that used hundreds or thousands of pairing, and my protocol had only 5, so it's much lighter procedure. And actually, I didn't show it, but I've measured also map plasticity using chronic intrinsic signal imaging, quantitative, and I did not see a drastic map change or expansion. There was a tendency to expand, but not significant. So the full answer would be when I do a more difficult task to this mice in the same. So I saw, I didn't show it. I saw that these mice can learn discrimination. So, but this is much more difficult for them. You need to do it over 10 days, very gradually. So it seems to be much more difficult. So I predict that after discrimination task, we would see map expansion, and then it will be really interesting to see what happens at the network level if I still have less or more neurons. And to totally complete the answer, usually the map expansion is transient, and the current model states that the role of map expansion is to increase the pool of neurons that are responding transiently, and then to choose from this bigger pool the ones that are most appropriate to the task. And then the map shrinks again, and you are left with the most appropriate neurons. So this is all my thoughts about that. Yes?
Well, thank you very much. Well, thank you for a very interesting talk.